Okay, so you're going to see this chart again. We saw it once before the exam, but when we're breaking down the nervous system, we want to be really clear that we remember that we have a central nervous system, we have a peripheral nervous system. So we have sensory information coming into the central nervous system, and then motor, efferent nerves going out of the central nervous system. So this whole slide set was on the central nervous system, and what we're moving forward into in this next unit is going to be stuff on the peripheral nervous system. So we've seen so far that the nervous system is going to involve nerves and also glial cells, and we saw a couple of them. So the central nervous system is the brain and spinal cord. And that's important to know because we're going to associate different parts of the brain and spinal cord with parts of our divisions of the efferent nervous system as well. Okay, so central nervous system, important for homeostasis, contains billions of neurons, trillions of synapses, so it's really big. It's responsible for everything. It's everything we perceive, everything we do, everything we feel, everything we think. It's forming these synapses that integrate all this information. That's also kind of poorly understood. So we know that it has neurons, that it has glial cells. It's got physical support. It's got blood supply. We looked at those in anatomy lab. And one thing that we didn't talk much about is something called the blood-brain barrier. So that's part of why I'm pulling us back here, is that the blood-brain barrier is really important, and I want us at least to see a picture of what it is and what that means. And we also have gray matter and white matter. We've seen that distinction, how it looks in lab, the difference between gray matter and white matter. But now that we know a little bit more about neurons, we can also understand on a sort of more histological level why we see that. Okay, so we've been talking about neurons. They respond to stimuli, right? We've been talking about them. Things we haven't discussed is that most of them can't divide. So most neurons, you can't really make new ones. You can repair them. They can make new connections to other neurons that they weren't connected to before, but they don't undergo the same cycles of cell division in the same way that a lot of the other cells in your body do. So they're kind of unique in that way. We classify them into groups. So we sometimes talk about their structure based on basically how many axons, dendrites, and stuff are extending from the cell body. We also can talk about them functionally, which is really what we focus on. So we got our sensory neurons, we got our motor neurons, we got our interneurons. We looked at that a little bit in physiology lab for those of you who have had the newest physiology lab. So we have our sensory neurons conducting information from the body to the central nervous system. We have our motor neurons conducting information about how we're going to react from the central nervous system to the target organs. And we have association and interneurons. We're going to look at a bunch of different types of motor neurons as we're moving forward. So I just want to be specific here that these aren't all types of central nervous system neurons. These are just ways we communicate with the central nervous system, except for these interneurons, which are entirely in the CNS. Okay. So we saw there are a couple different types of glial cells. We saw a little summary slide before. We can look at some pictures, but we're not going to focus on them. One that we will look at, the only one we're going to look at really more in detail here is the astrocytes. So an astrocyte has this star-like appearance. It has a bunch of different kind of appendages coming off of it. Appendages, we even name their ends feet, right? We can see these are end feet here. The reason we're going to touch on them a little bit more is that they develop connections between nervous tissue and non-nervous tissue, and they're really why we have a blood-brain barrier. It's possible they also modulate synaptic activity a little bit. We're not totally sure how that works, but they can communicate to the neurons through chemical messengers. So they're not excitable. They're not doing the same electrical signaling as neurons, but they do communicate. So we can see this communication happening here. Okay. Yeah. So they help maintain conditions in the central nervous system, and they protect neurons against toxic substances. So this again is going to be part of this blood-brain barrier concept that we're going to see in a second. Another cool thing is that they're involved with glycogen synthesis. And we'll see that a really important thing for the brain is to have constant availability of glucose. Okay, microglia, you know, they're important that you have them, but they're conceptually not that difficult. Conceptually, they're really just eating up bacteria, dead and injured cell. They're phagocytes, so they're doing endocytosis, breaking down materials that may be harmful for the brain. Okay. Glial cells are implicated in some neurodegenerative diseases. So we haven't looked at the glial cells yet that are responsible for myelin, except on that summary slide, but MS, multiple sclerosis, is actually due to the loss of myelin. So the loss of this protective substance around our nerves that allows action potentials to travel more quickly. As you lose that in nerves that should have it, you end up with some really weird leakiness kind of in those nerves that causes multiple sclerosis. Alzheimer's disease is going to be partially due to the loss of cholinergic neurons, and we'll get into what that means as we go into the autonomic nervous system. And it's where these neurons are really replaced by a plaque, which is to say a scar tissue. So that's damage to the actual neurons. Parkinson's disease is also due to loss of neurons, but in this case specifically dopaminergic neurons, so ones associated with dopamine. And in this case, the glial cells end up producing inflammatory agents, so inflammation in the brain surrounding also creates some of the things that we see in this disease. We're not going to talk about the physical support really because we've done this in lab, but we know there's bone, we know there are meninges, and this is really repeated here so that we can look at CSF. So we can see that we hopefully remember that we had these spaces in the brain, these that's not actually a picture of a ventricle, but we had ventricles in the brain that were filled with cerebrospinal fluid. Part of its job is as a cushion for the brain. So we will see that cerebrospinal fluid circulating between the brain and the bone. It's kind of released through this area. And that's involved with one of our glial cells. So our cerebrospinal fluid is created in the ventricles. It's this clear watery fluid that bathes the CNS. Note, we don't just see it surrounding the brain, we also see it surrounding the spinal cord. So when they do a spinal tap, part of what you're testing is the cerebrospinal fluid surrounding the spinal column. So I think they get at that through the sacral hiatus, which we've seen in lab as well. Okay, so that cerebrospinal fluid is secreted by ependymal cells. So that's our glial cell that I was getting to here. So those are in the choroid plexus, which is around the ventricles. So their job is to make the cerebrospinal fluid to cushion the brain. The cerebrospinal fluid also helps maintain important concentrations in this interstitial fluid, so maintains homeostasis there surrounding our neurons, surrounding our brain. Okay. These are just some concentrations. All you should note here is that concentrations of things in the plasma and the CSF are different. They're different fluids. Blood and CSF are not the same. Okay, so the blood-brain barrier. This is what I really wanted to get to. So the central nervous system counts for, to me, a surprising amount of body weight, but it's really a very small amount of your body weight in total. So it's 2% of the body weight, but it gets 15% of the blood supply. So in terms of its size versus its importance, like it's much more important than it is big. It also has a really high metabolic rate. So your brain uses a lot of energy, a lot of the oxygen. So 20% of the oxygen you breathe is used by your brain. It uses 50% of the glucose. So 50% of those sugars that we're metabolizing, right, through cellular respiration, they're going to the brain. So we're devoting a ton of resources to the central nervous system. It's going to depend on blood flow really critically, because all your tissues depend on blood flow, but also because these processes are going so fast, right? Because these processes in the brain, in the central nervous system require so much oxygen, require so much glucose, it's really important to have consistent blood flow because we're using up everything we get. Okay. So special things to know related to this, related to the blood and the brain here, is that neurons depend on aerobic glycolysis, which is to say, we talked about lactic acid fermentation. We talked about how muscle cells can still produce energy when there's no oxygen around. That's not true for neurons. Neurons need oxygen. Neurons need to go all the way through our aerobic cellular respiration process. So they really, really need a steady supply of both glucose and oxygen. If we run out of oxygen, we don't have a fallback plan. There's almost no storage of glycogen, so we also need a really steady supply of glucose. We don't have like a little glycogen bank hanging out there in the brain. Maybe a tiny bit in the glia, but it's not substantial compared to the amount of glucose your brain needs. Your brain also can't use fatty acids for energy. Using ketones is extremely dangerous. So basically, we just really, really, really need blood flow to the brain. And as we can see, really bad things happen if the brain does not get enough blood. Strokes are often caused by blockages, right, in blood vessels to the brain. So if you have a clot somewhere, travels and blocks blood to the brain, that's a stroke. This is caused by decreased blood supply. So that's why you actually see the things that you see when someone has a stroke. That's why you actually see them slurring their speech or whatever, whatever reactions you happen to see depending on the person. It's because the brain is not getting enough blood. So when we have occlusion of a cerebral blood vessel, so blood flow in the brain cut off, we see this, and there are also some strokes that are due to hemorrhage, so bleeding causing less blood flow in the brain. Okay. So the blood-brain barrier is really important because getting blood to the brain is really important, right? We're trying to get all that glucose and oxygen there, so we're rushing a bunch of blood to the brain, but we don't actually want everything that's in the blood to get to the brain. There are also a lot of substances in the blood that could harm the brain, could mess with our cerebral processes. So in most capillaries, where we have the site of exchange between blood and interstitial fluid, we have a thin layer of endothelial cells. They are joined by tight junctions, so those membranes are pressed against each other, but we'll see that in a lot of other places in the body, this layer of cells is a little bit leaky. We're going to allow some things to slip between the cells. We really don't want that to happen in the brain. So we do have some diffusion happening across the capillaries, and we'll see that in some places we're going to have like windows between our capillaries. So in the blood-brain barrier, our capillaries look a little bit different because we're really, really trying to limit that exchange. We want the glucose, we want the oxygen, but there's all sorts of other stuff that we don't want. That's the job of the astrocytes. Okay. So here we see a normal capillary. So here's what I mean. Our endothelial cells here in the wall of the capillary. That's just what the wall of a capillary looks like. These yellow parts, the cells. And we can see that the tight junctions would be like here, but sometimes in these corners, we're going to have pores in our capillaries. So if you slice it, right, like right here, you'd see that we have pores in certain places in the walls of this capillary. So we have blood here, I got our endothelial cells, but there's space between. So some solutes would be able to cross through these pores. We don't want this to happen in the brain. So our endothelial cells in those capillaries, they are a little tighter together. So we do clamp down on those pores a bit. But we also make sure they're tight, press them together, cover them up using the feet of these astrocytes. So we can see the astrocytes, those glial cells, are basically creating extra support on this wall of the capillary. So we don't see these pores. We got a tight junction here where we had a pore over here, right? So anything getting into the brain is going to have to go through those endothelial cells. So through a membrane on the blood side, then through a membrane here on the cerebrospinal fluid side, which means since it has to get through a membrane, it gives us the chance to control what gets through and what doesn't. So anything that we get into the brain is going to be carrier mediated, which means we're going to have specific proteins that help us get specific things across and only those specific things. Okay. So that was the real important thing that I wanted to get to, is just making sure that you know that a normal capillary is going to end up allowing a bunch of different things to cross because of these pores, but that's not happening in the brain, both because the capillary is stitched a little tighter and also because we have glial cells preventing it. Okay. The other important thing about glial cells, the two that we didn't look at, is that two of our types of glial cells form the myelin sheath. And we talked about myelin before, how that allows action potentials to travel more quickly. So we can look just a little bit at the formation of that myelin. So in the central nervous system itself, so like in the brain, the myelin sheath is made by something called an oligodendrocyte. Long word. If we break it down, I don't know, oligo probably means like connecting, dendro, similar to those dendrites, site, cell. So we have an oligodendrocyte. And basically what an oligodendrocyte is, it's a cell body here. So we see them in purple. It has many different processes that reach out to different neurons and wrap around. So the myelin is actually kind of a piece of these cells. When we were talking about myelin before, it's produced by and still attached to these cells, these glial cells. So oligodendrocyte sending extensions to many different axons, wrapping around sections, producing this myelin. So this coating is what allows us to have that saltatory propagation of the action potential, right? We only have to jump from node to node to node. So that's important because speed of conduction is important. And as we were saying, like with MS, if we start to not have as much myelin, then we start to get problems. This slide is specifying that it's not a neurolemma. We'll take a look at what that is in a second. But myelin is the thing that makes white matter white. So when we say we're talking about white matter versus gray matter, the white matter is going to be stuff that's coated in myelin. And that will come up a little bit as we start to talk about nerves out in the peripheral nervous system as well. Anytime you see gray matter, that's either cell bodies or it's parts of the cell that don't have myelin. Okay, so the neurolemma and myelin. Very similar to what's happening in the central nervous system, to these oligodendrocytes, but a little bit different. So when we're talking about a neurolemma, what we're really talking about is something called a Schwann cell, completely wrapped in many, many layers around an axon. So instead of having the oligodendrocyte with its many arms wrapping out to several different axons, we're like making a little cinnamon roll around the axon with our Schwann cells. We still have gaps here, so we have our nodes of Ranvier, but you can see each one of these little brownish tan pieces is itself a Schwann cell. So we can see that it has a cell body or nucleus, and then it's just a wrapping, coating many times around this neuron. So this is just to say that the myelin in the peripheral nervous system is a little different than the myelin in the central nervous system. Okay, and this is just looking at one. We already know that the point of myelin is axons can conduct those action potentials more quickly. Okay. Anybody have parts of this that they want to look at a little more, or are we ready to move on to autonomics? Okay. If you think of questions, we can always come back. Okay. So when we're talking about the autonomic nervous system, we're moving forward into the peripheral nervous system and specifically into its efferent division. So as we run through this autonomic nervous system, we're going to be thinking about the anatomy of the autonomic nervous system versus the anatomy of the somatic nervous system. So that'll be our next topic that we're going to be comparing it to. We're going to talk about the different chemical messengers and the different receptors that we're going to see in the autonomic nervous system. So that's going to separate partially different types of nerves in our autonomic nervous system. And we're going to be talking about two branches of the autonomic nervous system, which we'll call the sympathetic and parasympathetic branches. We'll see those in a second. And we're also going to think about how those two work together to create dual innervation. Finally, at some point, we'll talk about how the central nervous system links up to all of this and helps us control the autonomic branches. Okay. So this diagram again. So central nervous system was the brain and the spinal cord. Now, as we're moving into the autonomic nervous system, we're talking about the peripheral nervous system. So we're on this right-hand side, basically for all of this unit. So the efferent division of the nervous system, of the peripheral nervous system, efferent means we're talking about motor nerves, basically. So we're talking about sending impulses out to the body, telling our body to do stuff. This would be in contrast to the afferent division. Those are our sensory nerves. So those of you who have had phys lab already probably remember this distinction, but for those of you who haven't, this is what we're talking about. So efferent division, stuff going out. When we're talking about stuff going out, we can separate between the somatic nervous system, which is going to be going basically just to skeletal muscle. So it's going to voluntary parts of our body. So things that we can send voluntary impulses on, things you can think, oh, I want to move my arm, and then move your arm. And the autonomic nervous system, we can think of as kind of working in the background. These are going to be a bunch of automatic processes. Okay. So the autonomic nervous system, controlling these automatic involuntary processes, are eventually going to be affecting smooth muscle. So we'll see smooth muscle in the lining of our blood vessels, for example. We'll see cardiac muscle also being innervated by the autonomic nervous system. We'll see that glands are also the responsibility of the autonomic nervous system. So this will be tightly linked to like hormone production. Okay, so autonomic nervous system, unconscious stuff. You'll note that we have a division here, and I talked about the effector organs first because both our parasympathetic division and our sympathetic division are going to be controlling these things. So this division between the parasympathetics and sympathetics is not that they're controlling different parts of the body, it's that they have different effects on these same parts. Okay, so our afferent fibers, our sensory information, important for homeostasis, so we know when things in our body are changing. Our efferent fibers, our motor nerves, it's when we're doing something in response to some type of stimulus. Okay. So now we're thinking about these two branches of the autonomic nervous system. Sorry, scrolling back for a second. So we're moving from just sensory versus motor. Now we're going to be thinking about parasympathetic versus sympathetic. So we're skipping somatic until next time. Okay. So we're going to be describing the anatomy of these two branches of the autonomic nervous system. And then we're going to think about their chemical messengers. We're going to think about their regulation. Okay. So both parts of the autonomic nervous system are going to be innervating most of our effector organs. So like smooth muscle can receive both sympathetic and parasympathetic innervation. All right. The autonomic nervous system as a whole works kind of in secret. You don't consciously think about it. You don't consciously control it. We're going to separate parasympathetic and sympathetic. So you may have heard of like fight or flight responses. That's your sympathetic nervous system. So when we talk about a fight or flight response, what we're actually saying is that we're upregulating our sympathetic nervous system. We're triggering a sympathetic nervous response. So that's one part of our autonomic nervous system. The other part, the parasympathetic nervous system, is going to be responsible for the opposite. So our parasympathetic nervous system is responsible for rest and digest. I guess the digest isn't on there, but they rhyme. Your parasympathetic nervous system is going to be in control more when you're calm, when you're stable, when you're digesting, when you're at rest. So very important to realize that parasympathetic and sympathetic nervous system, they have opposite effects, right? One's in control of rest, the other's in control of fight or flight. But we really, really want to remember this first bullet point is that our organs are going to be getting communication from both our sympathetics and our parasympathetics. So while we want to associate the parasympathetics with rest and the sympathetics with fight or flight, we don't want to think about any specific organ as being the responsibility of just one part of the nervous system, because they're always going to be kind of balancing off. Okay, so we're managing a lot of different unconscious processes related to homeostasis using our autonomic nervous system, both our sympathetics and our parasympathetics. We can't maintain homeostasis without the autonomic nervous system, because it's controlling the size of our blood vessels, it's controlling our heart, it's controlling our glands. So we can't make the important adjustments we need to make for homeostasis if this isn't working. Okay, so here's a picture of our sympathetic versus our parasympathetic nervous systems. I'm going to pause for a second, and I want you to sort of make some mental notes to yourself about what information you can kind of glean from this picture before I tell you what I think you should be noticing. Okay. So first we're going to think about these organs in the middle. So you'll notice that what we've placed in the middle is parts of your body, right? We have the eye, we got the lungs, we got the heart, got the liver, we got the gut, all our organ systems here in the center. You'll notice the eye is getting some little green lines from the sympathetic nervous system. It's getting some little purple lines from the parasympathetic nervous system. So this is that idea about dual innervation, right? Any one of these organs is getting nervous input from both the sympathetics and the parasympathetics. So that's one thing that we can note here from this diagram. The other thing that hopefully you noticed has to do with the origin of these nerves. So when we're thinking about where these signals are coming from, we're actually thinking about different nerves coming out of the spine and brain. So here we have brainstem, and then here we have our spinal cord beginning. Okay. You'll notice that here for the parasympathetics, right, you got nothing coming out in the middle here, right? You just got a little bit at the top and a little bit at the bottom. So our parasympathetics we'll refer to as craniosacral, because the nerves that lead to parasympathetics in the body are either cranial nerves, as we've looked at in anatomy lab, or they're coming out of the sacrum, so at the very bottom of the spinal cord. All these pieces of the spinal column in the middle not contributing to parasympathetics. but that doesn't mean regions of the body in the center of the body don't get parasympathetic innervation. It's just that sometimes we're going to see this cranial nerve, right? It's our vagus nerve. Look how far down our vagus nerve goes, right? Our vagus nerve actually gets us all the way to the intestines, right? So all these different organs are going to have parasympathetics, but sometimes those parasympathetics are going to travel a really long way. The sympathetic nervous system also has its own place for the beginning of those sympathetic nerves. We'll see that we don't have sympathetic nerves coming out of the top of our spinal column. We don't have them in the bottom. We got them from the thoracic vertebrae and a couple of the lumbar vertebrae. So we discuss sympathetic innervation as being thoracolumbar because we're getting these nerves coming out of this central region. Yeah. Cool thing we'll also see, and we'll talk about in a second, is the sympathetic chain. So you won't see this in lab until second semester, I believe, because we haven't dissected deep enough into the body. But there is actually a physical kind of chained structure lying pressed against your vertebrae. So spinal cord inside, vertebrae surrounding. Just outside, we have this chain of ganglia called the sympathetic chain. It's big enough to be visible. Okay, so we're going to get into some text here now. Okay, so the two types of neurons we're thinking about as going from the central nervous system to any effector organ. We're going to have both preganglionic and postganglionic neurons. So when we're thinking about either our sympathetics or parasympathetics, we're going to be thinking about multiple neurons in a chain that synapse. And we'll take a look at those synapses and how they look in the sympathetic nervous system versus the parasympathetic nervous system. So we're going to see some differences in where they synapse. We're going to see some differences in receptors and neurotransmitters that we'll see associated with these preganglionic and postganglionic cells. So autonomic ganglia are the site of communication from the preganglionic and postganglionic neurons. We'll take a look at that. There are also sometimes intrinsic neurons that kind of modulate this connection. Finally, just a reminder, I've highlighted them when we're looking at diagrams, but you want to know that our effector organs for the autonomic nervous system, cardiac muscle, smooth muscle, glands, and actually also we now know that adipose tissue is an effector organ for the autonomic nervous system. You don't usually think of fat as doing a bunch of stuff, but it actually does stuff. So it counts as an effector organ. Okay, so here's what I was talking about with preganglionic and postganglionic. So when we were talking in our last unit, we talked about presynaptic and postsynaptic. It's a very similar concept. It's just here we're getting a little more specific that we're talking about a neuron before a ganglion and a neuron after a ganglion. So there are synapses here in the ganglion. So this is an autonomic ganglion. The reason we use a different word here is that we're not referring to just like the connection between one nerve and the next nerve. A ganglion specifically is somewhere you find cell bodies. So what we're saying when we say this is an autonomic ganglion, we're actually referring to the fact that these postganglionic neurons, their cell bodies can be found in these sites. So you'll actually be able to see ganglia in the body because they contain these cell bodies, which are, you know, they're not big, they're neurons, right? But they're big enough to be visible. Okay, so we're gonna have our preganglionic neuron traveling to these autonomic ganglia, and we're gonna have our postganglionic neurons traveling out to our effector organs. Okay. This is focusing here on sympathetics. So that's why we've picked that left-hand side of that big diagram. So our sympathetics are originate in the thoracolumbar part of our spinal cord. So here we got our brainstem, here we got our spinal cord. Here we can see the thoracic and lumbar sections of our spinal cord. Okay, so specifically, we're going to see that preganglionic neurons for the sympathetic nervous system lie in the lateral horn, and we'll take a look at where that is, which we can also call the intermediolateral cell column. Those terms are relatively interchangeable. Lateral horn is easier. Okay, so they're going to originate in a specific part of these, of this spinal cord, in the thoracolumbar spinal cord, specifically in this lateral horn. The general way that the sympathetic nervous system is going to work, so most of our sympathetic nerves are going to have a very short connection for our preganglionic neuron. So our preganglionic neuron is going to start here in the central nervous system in the spinal cord. It's going to leave the spinal cord out to the body. That axon is going to be short because it's only really trying to get to the other side of the bone to get into the sympathetic chain. Okay, so we'll have a short preganglionic neuron to the sympathetic chain. And then we'll have a longer postganglionic neuron out to our effector organs. Okay. So we got a preganglionic neuron, a little short step to the sympathetic chain. There we synapse with our postganglionic neuron. We got the postganglionic cell body there. Then we got a longer axon coming out for our postganglionic cell. So that's our typical, most common formation for the sympathetic nervous system. Another thing you might note if you look at this sympathetic chain very carefully is that all these ganglia are kind of connected together, like it's a chain of ganglia. So it is actually true that a preganglionic neuron can come into the sympathetic chain and then travel up or down it to synapse with a postganglionic neuron that's at a higher level or a lower level than our original preganglionic neuron when it entered. So that's just kind of a qualifier. It's not always super, super short. So some are short, some are a little bit longer, but we can kind of abbreviate for ourselves. So those ganglia are linked together. We do have some exceptions. So that's how most of them are working. That's the most stereotypical association for a sympathetic neuron. Sometimes we are going to have ganglia that are not in that sympathetic chain. So these ganglia will work basically the same. We'll have a preganglionic neuron coming out of the thoracolumbar spinal cord. It'll go to a ganglion synapse with a postganglionic neuron. But that ganglion, if it's not one of these in this chain, we call it a collateral ganglion. So we can see here that this nerve, right, this preganglionic nerve from L1, for example, you actually see that even though the line crossed over the sympathetic chain, it didn't do anything in there. It didn't really enter. It just continued out to this collateral ganglion. And that's where you see it actually connecting up to the postganglionic neuron. So that's where you see the synapse with the next step in the chain. So that's what a collateral ganglion is. So it works similar. It's just that the ganglion that they're going to, where they hook up with the postganglionic neuron, is not part of that chain. The bigger exception is what happens in the adrenal medulla. So the adrenal medulla is the central part of your adrenal glands, right? And the adrenal glands basically have a modified version of our sympathetic postganglionic neurons. So when we look at the adrenal medulla, it looks like these are just adrenal cells, which they are, but we can also kind of think of them as our postganglionic cells. So when we're looking at the adrenal glands, if we're thinking about the sympathetics, we'll have a sympathetic preganglionic neuron go all the way into the adrenal gland, into the adrenal medulla, and then synapse with a chromaffin cell. So a chromaffin cell is like a kind of super compressed postganglionic neuron. It gets its own name because it's special, right? It's in the adrenal gland. It's not really a neuron, but it's similar. Okay, so this should be the bigger exception in your mind, because this is kind of weird. Okay, but this is a thing that happens with the sympathetic nervous system. Okay. So in the adrenal medulla, we're going to release 3 types of catecholamines. So these are neurotransmitters. Some of them will also work as hormones, but we're thinking of them as neurotransmitters for right now. Okay, so 80% is going to be epinephrine. So 80% of what's getting released here is epinephrine. 20% is going to be something called norepinephrine. And then there's going to be also a very small amount of dopamine. Okay. Also, just a side note, we're using the terms epinephrine and norepinephrine here. Epinephrine is also known as adrenaline. So, okay, so here we see that all summarized in a picture, those 3 ways that we can have our sympathetic nervous system synapsing between our preganglionic and our postganglionic neurons. So here we see the bottom of the sympathetic chain. So here we see our preganglionic neuron coming out. And here you can see what the lateral horn is. So here's our lateral horn. So that's the origin of our preganglionic neuron in the thoracolumbar part of the spinal cord, so in the CNS. So we have our preganglionic neuron coming out. And here's the most typical, the stereotypical sympathetic nerve, goes into the sympathetic chain. That's where we find the cell body for our postganglionic neuron. So we synapse between our preganglionic neuron and our postganglionic neuron. And that postganglionic neuron goes out to our effector organ, maybe smooth muscle, maybe cardiac muscle, maybe a gland. Okay. Down at the bottom, we have our 2 exceptions. The one that's kind of like these sympathetic chain ones is the one where we have our preganglionic neuron go out to a collateral ganglion. It's just not in the sympathetic chain, it's somewhere else. We have some lying on like the aorta, for example. There are various ganglia lying there. Okay, so, but we're going out to a collateral ganglion, postganglionic to a neuron, sorry, to an effector organ. Then we have our extra special type of sympathetic nervous connection. We have our sympathetic preganglionic neuron going out all the way to the adrenal medulla, so to that central part of the adrenal gland. Then we have our chromaffin cell, which basically acts as our postganglionic cell here. Okay, so it's not a neuron, but it's basically an adapted version of a postganglionic neuron. I am being specific that we're in the adrenal medulla because we're going to talk about the adrenal cortex, so the outside, the surrounding layer, later. Okay. So this final piece, actually we've got a bunch of stuff about the sympathetic nervous system. I was hoping we'd finish up with this today. Okay. We're going to look a little more in detail about this pathway as we're thinking about our sympathetic chain. So our simplified version is just preganglionic, sympathetic chain, postganglionic. But there's a little more going on here. So that preganglionic nerve coming out of the lateral horn, very specifically coming out of the ventral root. Okay, so the ventral root, ventral is like your stomach side, dorsal is like your back side. Dorsal root is mostly like sensory nerves. So ventral root is where we would find our preganglionic sympathetic neuron. We're going out the ventral root from our lateral horn. We're going towards our spinal nerve. We're going to jump into the sympathetic chain, so into these linked ganglia via something called our white ramus. Okay, so here we can see that we're traveling in, going in our white ramus, white because we'd see myelin. Okay, and here we see the synapse, right, between our preganglionic and postganglionic neuron happening in the sympathetic chain. And then we see that that postganglionic neuron is jumping back onto the spinal nerve through the gray ramus. So we're thinking unmyelinated gray, right? White matter, myelin, gray matter, no myelin. So gray ramus and our postganglionic neuron is going out to our effector organ. So we can see how they're jumping on and off that spinal nerve in order to do this synapse in the sympathetic chain. Okay, so we will pick up here on Monday. I already said no quiz for the weekend, so that's good. Okay. All right, everybody have a great weekend. I hope you get some time to relax this time.